and I thought it was kind of interesting. Yeah, so first off, since it's Friday, we could all use a little bit of fun. I wanted to share a couple pictures of my kitten. Her name is Olive, and I've had her for about 3 months now. And if I'm working from home and in any meetings, you might see her running around my office, chewing on extension cords, that sort of thing. Okay, now to get into the topic. First, I want to talk about ethics because that's obviously very involved in experimentation. So, one of the earliest foundations of medical ethics is the Hippocratic Oath, which was written sometime in the 3rd to 5th centuries BCE. One of the important parts here is that I will abstain from all intentional wrongdoing and harm, especially from abusing the bodies of man or woman, bond or free. And of course, the Hippocratic Oath is usually included in the ceremony when people become medical doctors, but it's more of a moral obligation and isn't actually law anywhere. So it's you should do this, but there's not necessarily a punishment if you break this code or oath. Later in history, after World War II, they introduced the Nuremberg Code, which was created largely due to the outrage at the use of human test subjects in experiments, many of which were war crimes. Others included people using their slaves or children or people that could not properly consent to being experimented upon. And an interesting part of this code says no experiment should be conducted where there is a priori reason to believe that death or disability injury will occur, except perhaps in those experiments where the experimental physicians also serve as subjects. So basically, you shouldn't be experimenting on anyone in a way that could harm them unless you're also willing to be a subject in that experimentation. Again, this was not law, it's still just a moral agreement. Yeah, so there's some more modern documents ethically for medicine, one of which is the Declaration of Helsinki, which was initially written in 1964. It was developed by the World Medical Association, so it applies to all countries. It was based or built upon the Nuremberg Code, but is more specific to clinical research. And in some cases, they did relax the conditions for consent as some time had passed and we were doing more research and things like that. And I guess um, the idea was that we weren't going to be having any more war crimes, so they allowed proxy consent, such as a parent or guardian, and consent if at all possible, which did leave some wiggle room for medical research. An interesting thing about the Declaration of Helsinki, it seems to be updated every 10 or 20 years. And in the year 2000, they made some changes that the FDA did not agree with. So they have decided to go by good clinical practices instead of the Declaration of Helsinki, unlike a lot of the rest of the world. The good clinical practices were created as a response to unsuccessful and ineffective clinical trials. So they're more of a quality standard than like a moral and ethical standard. And they do lack some of the moral principles from the Declaration of Helsinki. They don't prohibit disclosure or yeah, they don't require disclosure of conflicts of interest. They don't require benefits for the populations used for research, and they don't really govern reporting of accurate results or negative findings in studies. So these are definitely more commercial practices that would benefit medical fields in that way, rather than necessarily benefiting the good of everyone. So I thought those were kind of interesting. And now let's talk a little bit about ethics committees and their thoughts on self-experimentation. So these are taken from a paper where they reached out to 37 different ethics committees, and they found that not all of them have policies on self-experimentation. 2/3 of all of these committees had no such policy. And I think it would have been more on a case-by-case basis rather than saying that, yes, self-experimentation is allowed. No, it's not. 2/3 of the committees also required an ethics review, but this was not tied to the ones that did have a policy on self-experimentation or not. So some may have had a policy on self-experimentation and required an ethics review. Some may have had neither, and combinations of the two. Some committees have suggested having a surrogate investigator to get informed consent from the scientists if the protocol is deemed to be too risky, just ensuring that everyone is aware that the person performing the experiment is aware of what could happen to them, rather than just expecting someone to say, yes, I know what will happen, but no one's going to look at that and deem if it actually is risky, that sort of thing. And one of the ethics committees considered self-experimentation to be meaningless due to the N of 1, which is a whole discussion for another day, I guess. Now I wanted to talk a little bit about the prevalence of self-experimentation in science, mostly in medicine. So about 465 self-experimentation papers were viewed in this review paper. As you can see here, they cover a wide variety of topics over just about 200 years. Most self-experimentation happens in the field of infectious diseases, anesthesiology, and physiology, whereas a lot less is going on in radiology and oncology, possibly due to the risks involved or just that it's harder to experiment on yourself with regards to cancer rather than infectious diseases. And the United States had the highest percentage of researchers performing self-experimentation, followed by Germany, the United Kingdom, and Russia. No other countries had a very high amount of researchers performing self-experimentation. Also shown here, almost all of the self-experimentation happened or a large majority happened in the early 1900s, and then it dropped by over a half after the Nuremberg trials and more regulations came into effect. So after all this, you might be thinking, why would I do self-experimentation? You can avoid the considerable use of time and money by testing on yourself rather than needing more rigorous approvals to test on other people. Usually it's a lot easier to get approval to do some small tests on yourself rather than others. And I think that's actually something the lab has experienced with like the Tasso blood devices. I believe we needed a lot less approval to do that than for Ashley to take the blood. So that's definitely something to consider. It's also cheaper and easier to do on yourself compared to a larger group. If we just needed one person's blood for that clinical trial, like I'm sure we could get that easily, but a large group of 20 is going to take a lot more time, a lot more coordination. It's also self-experimentation can be used to prove that it's possible to translate research from animal models to humans. You can try once. If it doesn't work, then you haven't wasted a lot of resources. And if it does work, then you can build on that self-experiment. And probably the worst reason, or the most risky reason, is to prove that something is safe, even if no one else believes you when you say that it is safe. So what are some positive outcomes of self-experimentation? Of the papers in this review, 98% of self-experiments had positive results, which either supported a hypothesis or provided other valuable data. And even if there were negative or inconclusive results, those could be used to direct future research without expending a lot of resources. And 12 scientists have received Nobel Prizes, most of them for the work related to their self-experimentation. However, there are obviously negative outcomes of self-experimentation. Going back to the trends in self-experimentation, most of which was taking place in the field of infectious diseases, there have been 8 deaths involved with publications for self-experimentation, and 8, almost all of those were from infectious diseases. One was not the most recent one, Alexander Bogdanov, who studied blood transfusions, and they believe that his cause of death was a transfusion from the wrong blood type, but he was the only one in this list that died from a non-infectious disease. However, there have been at least 190 documented self-experiments since the last death, and it has almost been 100 years since the last one, so I think things are definitely getting more safe. There's also a risk of long-term or severe injury. Jesse Lazier was a scientist who died from yellow fever while studying how it was transmitted, and one of his coworkers, James Carroll, also worked on that study. He did survive the acute yellow fever illness, but he never fully recovered and died just a few years after, much sooner than he probably would have if he had not been a part of that experiment. And I just wanted to talk about a couple of interesting cases of self-experimentation. This is one that I learned about in microbiology class, and it's the link between Helicobacter pylori and gastritis and peptic ulcer disease. So Barry Marshall worked with a pathologist interested in gastritis. They were able to culture Helicobacter pylori and believed that it caused gastric ulcers and cancer. Their first experiments didn't really show anything supporting their hypothesis. Their cultures weren't showing any growth initially, but they realized that this bacteria grows much slower than they were expecting. And the lab techs were getting rid of their cultures after 2 days due to prior protocol. But this bacteria actually takes much longer than that to grow. So they were able to finally culture this bacteria. And then they attempted to inoculate piglets with the bacteria to prove that they caused the gastritis, gastric ulcers, that sort of thing. But these experiments failed. After they failed, Barry Marshall had an endoscopy done and then drank a broth with cultured Helicobacter pylori in it. He expected to maybe develop ulcers years later in his life. However, 3 days later, he developed symptoms. Within a week, he had another endoscopy done and had the beginnings of gastritis. And 2 weeks later, he began antibiotics to treat those ulcers. They were able to culture Helicobacter pylori once again from his stomach and prove the postulates about the infection. And in 2005, he received the Nobel Prize in Physiology and Medicine for his research. Another interesting one I came across was cardiac catheterization. This man, Werner Forstmann, believed that you could put a catheter into the heart to more quickly deliver drugs, dyes, or to take measurements like blood pressure more easily. In 1929, he performed a surgery on himself, ignoring his department chief's warning with the help of an operating room nurse. She agreed to help him in the surgery only if he would insert the catheter on her instead of himself. But according to what I read, he strapped her to the table, anesthetized her and himself, and did the surgery on himself while she couldn't do anything about it. And then, so yeah, he inserted a urinary catheter through a vein in his arm, and with X-ray help, he was able to guide it into the heart, and then they took the X-ray to prove that it was actually in the heart, which is the image you see here, the little black line going beneath the bones in the arm is the catheter that he placed in his own heart. He was awarded the Nobel Prize in Physiology and Medicine in 1956, but he did have a hard time getting a job after doing this because the hospitals didn't want to hire someone who was just going to experiment on themselves against what they had told him. So he had a few years where he wasn't able to work despite doing something that was so useful that it got a Nobel Prize. Another category of self-experimentation is non-infectious diseases. This is more the category of proving that something won't work rather than proving that it will. I think they're still pretty interesting. So Elliot Cutler is a researcher who took thyroid extract in order to give himself hyperthyroidism so that he could study its effect on his body and his kidney function rather than finding someone with hyperthyroidism who would agree to his experiments. In the 1700s, William Stark was attempting to find the cause of scurvy, and he had tried 24 different dietary experiments and was taking measurements of how much he ate, how much he excreted, as well as environmental factors like the weather and the temperature to see if he could figure out what caused scurvy. He first started out eating a diet of just water and bread, but declined physically because of that. He then tried different combinations of food and just developed scurvy within 2 to 3 months. However, he died before he was able to try a diet with fruits and vegetables in it, which are what has been proven to prevent scurvy. So he started on the right track, but did not survive long enough to come up with, I guess, the answer there. And there are also several instances of scientists who have injected themselves with cancerous lymph nodes, discharged from cancers, or sterilized cancer extracts, and they have not developed cancer from this, showing that cancer is not an infectious disease. Another area where people have done a lot of self-experimentation is with various drugs and chemicals. Scientist Edwin Katsky in 1936 took a large dose of cocaine. He was writing notes about what was happening to him while he was, overdosing on cocaine. An excerpt here is to the side. He talks about how these results will be recorded. He needs to have a college look at them because he's not going to be able to return or to repeat the experiment. I think the last note he allegedly wrote was paralysis, and then when they found him the next morning, he was dead with this notebook beside him. Another scientist, David G. Bailey, was testing the effects of alcohol on an experimental drug felodipine. The protocol was to test with alcohol mixed with orange juice, but he didn't like the taste of orange juice, so he mixed the alcohol with grapefruit juice, and from this accidentally discovered that grapefruit juice affected the metabolism of the drug when he had 3 times the amount of the drug in his blood the next day compared to the rest of the scientists. And it was later shown that the grapefruit juice affects the CYP450 enzymes and affects metabolism of many drugs in the body. Another scientist was Albert Hofmann, who was studying psychoactive drugs and was the one to initially synthesize LSD. He absorbed a small amount of it through his skin and felt some effects from it. And the next day, he decided to take a much larger dose, which I believe is like 100 times more than is necessary to feel any effects. He then attempted to ride his bicycle home while having the world's first LSD trip, and this is known as Bicycle Day. He wrote down his experiences of this novel psychoactive drug. And yeah, it was certainly not the amount he should have been taking, but it was, yeah, good, interesting research to be doing on yourself. There's also a couple of cases of unintentional self-experimentation that I came across, one of which is James Schlatter, who discovered aspartame while he was synthesizing a gastrin peptide. It ended up being one of the metabolites in the synthesis. He had licked a piece of his lick he had licked his finger to pick up a piece of paper and noticed a sweet flavor, which was the aspartame. But I don't think this is something we should practice in the lab. Other scientists who did unintentional self-experimentation include Nikola Tesla, who experienced burns on his skin and eyes from his experimentation with X-rays before they knew that there were any risks, as well as Marie Curie, who died of anemia, which was likely caused by long-term radiation exposure. So I guess overall, there's a lot of good things that have come from self-experimentation, probably a lot of money saved, a lot of research that's gone forward because of this. But you have to be careful to fully know and understand the risks of what's happening, and to rely on ethics and whatnot when you are performing research like this. Yeah, so that's my presentation. Here are the references I got the information from. Yes, does anyone have any questions?
Nicely done, Eponine. Very informative.